Yeah, our research is on DNA damage. We work at NYU in the Stowaway Center with Professor Nicholas Lucentoff and Dr. Jay Gu. Okay, this is our outline. We're going to start with some definitions, and then our background research, objectives, materials, experimental section, results, future work, and then acknowledgement and work. So here's some basic definitions of what's to come in our PowerPoint. DNA is basically, it stands for deoxyribonucleic acid, and it contains the genetic information of the cell. A buffer is a solution that allows to maintain a pH. Something that's acidic has a pH below 7, and something that's basic has a pH above 7. The absorbance is the measurable amount of light absorbed by a sample as light passes through it, and you measure that with a spectrophotometer. And Bayes' law states the absorbance is directly proportional to the concentration of a solution, and that's the formula for Bayes' law. Something that's carcinogenic is cancer-causing. DNA adducts form when a cancer-causing compound binds to the DNA and thus modifies it. A lesion is a DNA damage base and nucleotide excision repair, also known as NER, the process of DNA repair where damaged sites called lesions are recognized, excised, and replaced. This is our background information. There's a large number of compounds that cause mutations in DNA, and that's basically what we're researching, what causes that. Benzopyrene binds covalently to double-stranded DNA to form adducts, and DNA adducts give rise to lesions in mammalian cells.
So the objectives of our study is to find out how these carcinogenic products basically change the base sites and what are the damages that are caused by the changes in the base pairs. So in our specific project, we're going to be studying the behavior of the chemical written over there, which is benzopyrene diol epoxide derived DNA adducts, which are damages caused by the chemical mentioned. And we basically study the changes that happen to the DNA and what are their biological properties, how their function changes, how the proteins that are transcribed by the DNA, their structure changes and their function changes basically in the human body. So till now we've just started doing it, so this is like a training session. So basically what we did, these are some of the materials, the instruments and the chemicals that we use for our training, the research.
And the top picture is the spectrophotometer that measures absorbency.
Okay, so our first experiment was to check for pH measurements. So to basically we used different concentrations of sodium phosphate monobasic and sodium phosphate dibasic. We added them together and we measured at different concentrations what their pH is, like different concentrations of monobasic and different concentrations of dibasic were added, and to see how their pH of the solution changes as we move along. So this table basically summarizes our whatever we did, like how much concentration of the first chemical was added to the second chemical. So, and we finally, after we made the solutions, we basically measured the pH and put it into a graphic format. So this was our results. So basically, as we did one of the chemicals, when we did the dibasic, we can see that as the concentration of the dibasic compound added increases, we can see that the pH is going up. Okay, so our second experiment was to make like a DNA buffer solution. This is important because when we are doing our research, it is important for DNA to measure, to be maintained at a constant pH, because in your body too, you have your DNA at about 7.4 pH in your blood. So that's important to maintain it while doing the experiment. So to make a buffer solution, we added the 3 chemicals in their respective quantities. So we were given a specific concentration, and then we had to find out how much amount of the chemical that we needed to have. So we took a common volume of 50 ml, and we basically found the amount that needs to be added into distilled water by using the formula concentration equals moles over volume. And then we added all 3 substances into distilled water, and then we measured the pH. This turned out that the pH of the buffer was about 6.92. It should be about 7, but it's with errors accumulated, so anywhere between 6.8 and 7 is fine.
And that was the second buffer you made.
Our third experiment was to study absorption measurements. This is important because while studying DNA, to study how much damage of something has happened, you need to use spectrophotometers to basically measure how the absorption has changed. So, for example, a damaged DNA might absorb light at a different wavelength than normal DNA. So we had to learn how to use the spectrophotometer. So for that, we basically used a compound called 2-deoxyguanosine, and we added that to 1 ml of distilled water. And we took the buffer solution that we previously prepared, and we measured various quantities of the deoxyguanosine and added it to the respective quantities of distilled water and buffer solution. In the next graph, as you can see, we have been increasing the concentration of deoxyguanosine, and as we increase it, the amount of distilled water that we had has also changed, but the buffer solution remains constant. And then after doing that, we got like 10 solutions, and we basically put them inside the spectrophotometer and measured their absorbance. So these were our results. So earlier you guys saw about the Beer's law. Basically, this illustrates that principle. As you can see, the concentration keeps increasing, the absorbency also goes up.
Yeah. In the future, we're going to study the DNA duplicates in the coming weeks, and we're going to use UV absorption methods to do that. But we haven't really seen the materials for that yet, but coming up, we're going to do it. We'd like to acknowledge Professor Nicholas Crescento, Dr. V. Wu, NYU Chemistry Lab staff, Dr. Chat, HGS staff, and all of you for listening. The references.
Any questions? Yes.
So you guys are trying to improve the conditions
That's not what we're trying to prove, but that's how we're going to find out if something is damaged or not. It's just a step in between. So we are basically trying to see how the specific damages cause, problems in our human body.